said, I'm from the Pulmonary division rather than the Allergy division, but of course, my research mentor is Bill Hallstrand, sitting right here, when I started down this road, so he's responsible for anything that you might criticize about my work. But I thought I would basically talk about mast cells and asthma, and a lot of this is really what we found. I'm also going to talk about secretory PLA2s and how they might regulate allergic inflammation as well. And I'll be talking about a couple of projects that we have ongoing. One is the project that Sydney Long is working on, and then another project, Jimmy Nolan and others in my lab, where we're doing some mouse modeling I'm working on. So I have no disclosures. The objectives are to consider the heterogeneity of asthma, recognize the relationship between mast cell infiltration and exercise-induced bronchoconstriction. We'll talk about kind of what that means, and appreciate the function of secreted PLA2s as regulators of type 2 inflammation. So the key points that we'll come to today is really that exercise-induced bronchoconstriction is a distinct feature of asthma. Mast cell infiltration of the airway epithelium is characteristic of people who have this disorder. Th2 high asthma, which has been developed by the research group at UCSF, is really, as it turns out, a marker of both indirect airway hyperresponsiveness and mast cell infiltration. And then epithelial-derived secreted PLA2s may serve as a key regulator of lipopene formation. So this is really what got me interested in this area, is that I started to study this disorder, which is exercise-induced bronchoconstriction. Or is it on the page? Oh,
Is it on white?
So what you see is that this is lung function measured on the side there. So FEV1, lung function, and before exercise, and then you do a standardized challenge test. This is both kind of what happens to people in the real world when they go out and exercise and they reach a certain threshold, but this is also a standardized challenge test that we use in the lab called the dry air exercise challenge test. And so if you do that exercise challenge test, which is basically standardized by using 6 minutes at either a minute ventilation that's up in the 40 to 60% of maximum, or your heart rate up to around 80% of maximum. For 6 minutes, that serves as the maximum trigger, and then you develop bronchoconstriction as shown there. That'll last for somewhere between 30 and 60 minutes. And this is just kind of showing what happens in the airway. So this is a hyperpolarized helium image. So it's basically like a CAT scan, except this is gaseous contrast, so hyperpolarized helium. And here it is post-exercise. This was a study done by Tom Platzmels. And so what you can see is that there's whole areas of the lung which have no ventilation, and then there's big segments of the lung that have heterogeneous ventilation, and that's consistent with closure of the airways or near closure in the conducting airways where this disorder is thought to occur. So the first point is really that this is exercise-induced bronchoconstriction is a distinct feature of asthma. In cross-sectional studies, like when I started, I was told that basically everyone with asthma, if you go out and exercise, will trigger and develop exercise-induced bronchoconstriction. But as it turns out, in cross-sectional studies, somewhere between about 30 and 50% of asthmatics will actually trigger to that dry air exercise challenge. And you can see from the graph on the left, looking up that this occurs. Looking at lung function responses in all levels of severity. So you can identify people that are mild asthmatics that do and do not have exercise-induced bronchoconstriction. You can go all the way up, and the same thing is true at all the way up into severe asthma. And it doesn't seem to be very strongly related to what your baseline lung function is. So this is showing baseline lung function versus the drop in lung function after exercise. So it doesn't seem to be a marker of structural changes in the lung. But what it does seem to be a manifestation of airway hyperresponsiveness. So when we measure hyperresponsiveness in the clinic, we measure that with a test called a methical methicillin challenge. And so a methicillin challenge is a classic example of what we say is direct airway hyperresponsiveness. That means it's just binding to the airway smooth muscle and causing constriction, and we basically do a dose response. And most people kind of say that if you have asthma, you should have a positive methicillin challenge, and that's largely true. I'm actually on the bronchoprovocation task force, the people that are rewriting the guidelines for how you do this testing. But there are several studies that suggest that at least in periods of quiescence, patients with asthma might have a negative methicillin challenge. So if you look at people that have definite direct airway hyperresponsive to methicillin, and then you test them to exercise, about 60% of them will trigger. So there's some cross-relationship between direct airway hyperresponsiveness and hyperresponsiveness to exercise. Exercise, in contrast, is considered one of the stimulus in which we call it indirect airway hyperresponsiveness, because you seem to have to trigger a mechanism that works indirectly through other mechanisms, which we'll kind of go over. So if you look at it in another way, if you just take people that have exercise-induced wheezing, they report that symptom, about 60% of those will have a positive methicillin challenge, and only about a third of them will actually have exercise-induced bronchoconstriction. And so one of the problems in this area is that reported symptoms don't really tell you whether or not someone has this disorder very well. And a lot of those people have other disorders, such as vocal cord dysfunction. And then if you look at it in yet another study, this is one of a couple of studies that show this, that at least in some people, it's possible to have a positive exercise challenge. In this case, a test called a eukapnic voluntary hyperpnea challenge, a surrogate challenge, and not have a positive methicillin challenge. So it seems like this disorder is related to airway hyperresponsiveness, but it's distinct in some ways. So we usually think of it this way. And so that if you have asthma, you usually should have a positive methicillin challenge. Among those people with a positive methicillin challenge, there's a subset that have exercise-induced bronchoconstriction, or EIB. If they have a negative methicillin challenge, you should really consider other diagnoses. A lot of people turn this question around the other way because athletes, of course, report lots of symptoms like this. And some athletes will have EIB. The vast majority of those athletes will have a positive methicillin challenge, but at least in some series, that there's some of those people who actually have a negative methicillin challenge. So if you suspect it clinically very strongly, a methicillin challenge by itself might not be enough to really confirm the diagnosis. So we became very interested in why this occurred in this subset of patients with asthma. And so if you look at the table over there, you can see that we were able to enroll two groups of asthmatics that do and do not have exercise-induced bronchoconstriction. So they look identical as far as their lung function and bronchodilator response, but their phenotype, when it comes to their response to exercise challenge, is markedly discordant. So it allows you to kind of hone in on the one endotype of asthma, essentially. And what we found was that when we looked at their airways, in this case using induced sputum, that they had a lot more epithelial shedding into the airway lumen and higher levels of leukotrienes, and particularly a high level of leukotrienes relative to prostaglandin E2. So those eicosanoids, so leukotrienes cause bronchoconstriction, and PGE2 is kind of bronchoprotective. So once you get that ratio seems to be markedly altered. So this led us to do further studies that were meant to tease out what was different about the subjects that had EIB, or more generally indirect airway hyperresponsiveness. And in this case, we did a high throughput gene expression study. And this was really actually the first in which we applied this to induce sputum. And we had to use a number of techniques to basically stabilize RNA so that we could do this adequately. And so again, we had these two rigorously characterized groups. And there were a number of genes that were differentially expressed. And what this is showing is genes that were differentially expressed on the array, which is how we did it back then, prior to RNA sequencing and things like that have become more common recently. And then we did a confirmatory assay with a PCR, and this is looking at the log-fold difference between the two groups. And so these are genes that are differentially expressed between those two groups. And prominent among them are these two genes. So that's tryptase, and this is CPA3. And so they're both mast cell genes that came up as being increased in the group of subjects with EIB. About that same time, really, Prescott Woodruff and John Fahey's lab really has started looking at epithelial brushings in asthma to identify, again, using high throughput gene expression methods, differences in epithelial gene expression between asthmatics and non-asthmatics. So in this case, unlike what we did with two different phenotypes of asthma, this is asthma versus non-asthma. And this led to the classic finding, and I don't think I really highlighted it here, is of these three genes, which are now considered kind of the sentinel features of what's termed Th2 High asthma, Serpin B2, Periostin, and Clica1. And that's, again, just showing differences, full differences, and based on the array and then by PCR. And the other thing that they showed about this is that in vitro, when you cultured epithelial cells and you treated them with IL-13, these three genes also came up. And it seemed like the people that had Th2 high asthma were the ones that seemed to have had a positive response to corticosteroid therapy in the past. And so this led to kind of this paradigm that these three genes were increased. But what you can see just above that is that tryptase and CPA3 are also there, just looking at overall, but less differentially expressed. So they went forward from this study and looked at mast cell genes more globally in a follow-up study. And so this is actually a subset of patients that were from that original array study. They were just taking samples out of the freezer. And so this is looking on the array, and so showing that overall, in asthma, there's an increase in tryptase and CPA3. This is in epithelial brushings. I think the intent was not to look for leukocytes necessarily at first, and lower expression of kinase. And they showed that this was also true by PCR. So this showed kind of something, it suggested something interesting that people hadn't really thought about too much, that there might be mast cells in the epithelium that might be driving this. And the second thing is that mucosal mast cells, the ones you see in the epithelium, are typically only positive for tryptase, whereas mucosal mast cells are usually positive for tryptase, but also a wider array of other proteases such as CPA3 and kinase. And so these mast cells actually also have a funny intermediate phenotype because they have high expression of CPA3 but low expression of kinase. And so the mast cells themselves seem to have a phenotypic alteration. So then, because of that possibility, they went back to tissue samples from their lab, and they used a technique called design-based stereology that I'm going to talk about in just a second, to quantify the number of mast cells in the epithelium. And this is really one of the first studies. There's only a few studies in the literature total that use this technique. And this is basically a quantitative morphometry technique, and I'll describe it in a second. But you'll notice they're showing the number of mast cells per volume of epithelium in human subjects, which is really kind of revolutionizing because people before that had only looked at little cross-sections, and there's an inherent bias in that. But as you can see, unfortunately, in contrast to their initial hypothesis, the number of mast cells in the airway epithelium actually weren't very elevated. There's just a lot of heterogeneity and a trend towards an increase. But then they subdivided it based on that Th2 high and Th2 low asthma and compared that to control. And as it turns out, the people with the mast cells in general had Th2 high asthma. And so in a kind of post hoc comparison, they were able to show that there was this difference in the number of mast cells in the airways. So our second point then is going to be about mast cell infiltration being a key feature of that's what we studied directly. This really, what led us to this hypothesis was actually prior to the study by Foggy and Woodruff and everybody, was from our Array study, and we looked at these different groups here. And so here, in this case, we now have a control group, but we also have an EIB negative group and an EIB positive group. And you can see how they're phenotypically very, very distinct. Their baseline characteristics are not very different, including their methacholine responsiveness. So during exercise, so they have this 6-minute exercise, dry air exercise challenge test. You can see that they have differences in their symptoms. This is the Borg scale shown there. So the group with EIB is already developing symptoms, early on during exercise, and those symptoms, of course, persist because bronchoconstriction persists for like 30 minutes after exercise. And so in this study then, we did research bronchoscopy at baseline. So we looked at, we had each of them come in for a bronchoscopy. We did epithelial brushings, as shown here, and we did copy number PCR looking for the different mast cell genes in the epithelium. And I think I should have colored my slides differently because I think the pointer's kind of hard to pick out here. But what you can see is that that tryptase and CPA3 are definitely increased in asthma, but you can also appreciate right off the bat that the EIB negative patients really don't have a difference in the expression of either of those genes. It's all in the EIB positive group. So really the mast cell infiltration story, at least as far as you can infer it from the epithelial brushings, is in that one group of distinctive group of asthma with exercise-induced bronchoconstriction. Interestingly enough, we also found that kinase, shown there, is much lower, but it is slightly elevated in the EIB positive group. We looked then to see whether or not we could see the same difference using induced sputum the way we had done before. And you can see that largely, actually, you can use a relatively less invasive test, induced sputum, to show the same results. So there's a difference in the expression of these mast cell genes in the induced sputum. And then, remarkably, there's this very, very crisp association between how much your lung function falls after exercise and the gene expression for these mast cell genes in the airway epithelium, especially for tryptase and CPA3, but not so much for kinase. So we, at the same time, did airway biopsies in these patients so that we could do quantitative morphometry, and we used this same technique called design-based stereology. And I just wanted to take a few minutes to kind of talk about it. So stereology is basically a statistical science, and the basis of it is really sampling. And so you have to do random sampling in the airways. You have to have random orientation because it doesn't make assumptions about the orientation of the tissue. Like oftentimes in many studies, people would take a piece of tissue and then line it up in a specific axis, but there again, you have an inherent bias that you're introducing into your sample. And then you basically use a series of probes that allow you to assay for like the length, the size, and so forth. And so then you can define the length or volume of something relative to a reference space. And believe it or not, the American Thoracic Society and the ERS recommend that all studies, quantitative morphometry studies, should use these techniques. And the interesting thing is that only 2 labs that have published anything on this are John Pahey's lab and our lab here in Seattle. And John was the one who taught me how to do this initially. So, but you basically are using a series of probes. And so like if you want to know the length of, say, a nerve in the airway, once you have random sampling, you can put a series of planes in the, what's called the optical dissector, and then you could identify the length of the nerve by how often it crosses those series of planes. And the same thing, you can get a volume of a sphere by essentially creating a box. The reason that stereology is important is if you were to take just a random cut across, say, that series of spheres, and you're interested in the number of mast cells, depending on where you slice it, if you slice across where that larger sphere is, you know immediately that you're more likely to hit that large sphere than you are to hit the small sphere, right? And so if you're counting the number of something, you can see immediately how you have bias if you only just take a single section like that. So that's how stereology works. So if you do this in mice, it's pretty straightforward because you can take the lung out and then do a series of cuts. And so then you can take sampling out of those series of cuts, and then you can randomly orient the cuts, and that allows you to do what you need for stereology. In people where we're doing it, then it's a little more challenging. So we basically take 6 airway biopsies, and to take a biopsy, you have to basically get it off of a carina, so a branch point, because the biopsy forceps won't seat otherwise. And so, but we're interested in the conducting airways, and you're just kind of reaching those when you get into the 4th and 5th generation airways. So we basically go out to the 4th and 5th generation airways and take a little piece. And so that piece looks like this. This has airway epithelium on the outside. It's kind of rolled back up. That's actually stained for mast cell tryptase. It's showing that all those little dots in there are mast cells. And then we take those individual pieces. They're placed in what's called an Eisector mold so that we can randomly orient them. And then they're randomly oriented into a paraffin block. And so if you want to get at the density of mast cells, you put basically 3 of those tissue sections 3 microns apart. And then in these, you'll have to line them up so that you can count the number of mast cells that come in uniquely within 2 different sections that are a known distance apart, which is what gives you that 3-dimensionality to it. So the number of mast cells, the area, what's called the counting frame, the height, which is the difference between the 2 sections that you look at, and then you can identify the reference section with the points hitting the epithelium. So this shows how you do it. You have to line up the sections, and so you have a reference and a lookup section. And so you can see that these 2 sections are lined up, but they're 3 microns apart. You can see that there's here in the I can really point, but you can. So basically, this is what's called a counting frame, and there's rules of what you use to count something in or out. And so basically, you're looking for something that uniquely comes into the counting frame in each direction. And so, like, there's a mast cell here, a mast cell nucleus that you don't see on the other frame. And so that's a mast cell that uniquely comes in. If the mast cell is actually in both frames, it's not counted because it's not uniquely entering the frame. And then you can see and then you have points hitting the epithelium, and that allows you to do this.
How big is a mast cell?
As it turns out, mast cells are something like 6 microns. And I think this is actually wrong. Am I right about that? So this is we use a 3 micron dissector. So this is, so the Fahy paper that you saw, actually, they did use a 6 micron dissector. So they went from the first to the outside, and they counted mast cells in general. We found that was inaccurate, actually, and if you look in their study, they have twice as many mast cells as we have. And that's because if you look at these sections, there's mast cells that are broken apart, and it's really hard to tell if you just have a mast cell that exploded there, or it's a unique thing. So we went back to the literature and got advice from Dallas Hyde and other people in this area, and so we ended up counterstaining, so we'd have very crisp nuclei. And so we basically said, that's a mast cell, and there's a unique nuclei associated with it. And so I think we have a very rigorous number by doing that. So we counted mast cell nuclei, but because of that, we decreased the dissector height. So the second very interesting way of doing this is that you can do a different reference space. And so we did the surface area of the basal lamina. So the basal lamina is always altered in patients with asthma. It's that thickened area below the epithelium there. And so basically you can use that to get the surface area of the airway and how many mast cells are above it using a similar approach. So here's some staining for tryptase and carboxypeptidase A3. You can see mast cells that stain for both of those are up there in the airway epithelium up on top. And here's the stereology assessment. This is the number of mast cells per area of epithelium. And you can see, with the exception of 2 outliers in the EIB negative group, that all of the increase in mast cells is in this group that has exercise-induced bronchoconstriction, really demonstrating that phenotype. A really kind of defining feature is this mast cell infiltration of the airway epithelium. This is looking at it with that other method, looking at the basal lamina. So this is the surface area of the basal lamina. So the idea is that we can take out other factors like epithelial disruption and just look at what's above the basal lamina, and we've got the same result. And these are really markedly different, much more different than the Th2 high, Th2 low that I showed you before from John Fahey's study. And then interestingly enough, we looked at the volume of the epithelium relative to the basal lamina using stereology methods. And this has been reportedly abnormal in patients with asthma before, but you can see once you take out the bias, the volume of the epithelium is actually not altered in asthma. So this led us more recently then, so it seems pretty clear that mast cell infiltration of the airways is a defining feature of this group of subjects with exercise-induced bronchoconstriction, and exercise-induced bronchoconstriction is kind of like a hallmark indirect airway hyperresponsiveness feature. So we wanted to look at the overlap with Th2 high asthma. And so you could ask yourself, well, how are you going to do this and how are we going to integrate it? So this is a study from Mike Peters, and this was in JACI. It was actually about the same time as our mast cell paper. And they were interested, after doing epithelial brushings all this time, to go back and use induced sputum, much like we had done in some of our initial studies, to see at whether or not they could find a non-invasive measure of their Th2 high asthma. And so they first looked at the 3 genes that they had seen in the epithelium, periostin, CLICA1, and serpin V2. Now, the problem was that it really does seem a little bit more epithelial-centric, and so there wasn't a difference in serpin V2 right there, and there's quite a lot of overlap, although the CLICA1 seems to be the one that's the most different. Actually, if you looked back on our gene array paper that I was showing you, one of the other genes that was most differentially expressed was also CLICA1. So then they went in and looked directly at type 2 cytokines. If you've been following the literature, of course, people used to call everything Th2, and of course that's been dropped as people have realized that other cells, particularly innate lymphoid cells, are actually the key source of IL-13 in the airways, and so everyone has turned that terminology now to really type 2. But really the Th2 has kind of persisted. So, but anyway, so they looked at, and they use a little bit of a different protocol. They use a lot of preamplification with this huge set of primers. So they basically put a series of primers in there. They preamplify and then look at IL-4, 5, and 13. And you can see that these look like they're increased in asthma. And then they developed this metric. And to do this metric, you have to center and scale the expression for each Th2 cytokine. Then you calculate a mean across the 3 of them, and then they use that to classify these patients. So in their study, and again, these are really kind of banked samples from the UCSF program, they showed that there's this gene metric that gene metric is based on the healthy controls there of their own sample. So 2 standard deviations from the mean of the healthy controls. And you can see people that are not on inhaled steroids, some of them are Th2 high and some of them are Th2 low. That idea that it's a marker of corticosteroid response, I think, is what a lot of people think about it, but there's actually not much evidence of that here because even more people that are Th2 higher are on inhaled corticosteroids. So you can identify these different groups. So we sent them, and like I say, this is the project that Sydney's working on. We sent them our samples from that same cohort that we had. So first of all, just looking at IL-13, once you scale it, and then looking at the 2 standard deviations from the mean and the healthy control, that's the line there, you can see that especially patients with the EIB high kind of phenotype are more likely to have higher levels of IL-13 based on gene expression in their airways, although there's a lot of overlap. So if you then formally generate that metric of combination of IL-4, 5, and 13, you can see that in general, again, the Th2 high is more prominent in those people that have EIB. But the interesting thing was that in their studies, actually in all of their studies, they really haven't related Th2 gene expression to a strong phenotype other than this concept that it's somehow a marker of steroid response. It was since they've had larger studies, it was weakly associated with lung function, interestingly enough, but not very strongly. And the slope of the curve is very flat. And so in this patient population, then Th2 gene mean is not associated with lung function at all, but it is very kind of associated with methicillin hyperresponsiveness, but that's really driven by the fact that we have a bunch of our healthy controls over there with a PC20 of 8, a negative methicillin challenge that's driving the slope there. But a nice tight relationship, however, is between the Th2 gene mean and the severity of exercise-induced bronchoconstriction, and that's using, rather than FEV1, that's using a summary measurement called the AUC30 in this case. So it seems like the Th2 gene mean is reflecting indirect airway hyperresponsiveness predominantly. Using these samples, and again, they did the gene expression, so it was a repeat measurement of that. The CPA3 gene expression is also associated with the severity of EIB, which is what we had found before. And if you look at that Th2 gene metric and the number of mast cells per epithelial volume done by Stereology, you can again see that there's this really nice relationship. So it looks like the Th2 gene mean is really a good marker of mast cell infiltration of the airway, and it's a good marker of indirect airway hyperresponsiveness. So the future directions are really some of the things that Sydney's working on using Stereology. She's getting good at lining up these and counting mast cells. We're counting the number of mast cells in the submucosa, and of course one of the key questions here, because there was, Chris Breitling had a study on this, mast cell infiltration of the airway smooth muscle, which is what you're seeing right here. And so we'll be quantifying that as well. I don't think I'll be able to do what Chris Breitling did, which was, I think it was a subject, 13 subjects. It was in the New England Journal of Medicine. And then, of course, there's been a number of studies that have since contradicted his original finding. But I would imagine that potentially because we have so much more crisp phenotypic information in our study, then we'll have more chance of resolving what that relationship actually is. And we'll also be looking at eosinophils. As it turns out, there's also a modest relationship between airway eosinophils and these same findings. So we have this model then that these patients with exercise-induced bronchoconstriction have disruption of the airway epithelium, infiltration with leukocytes, particularly eosinophils and mast cells, and that leads to these findings of high leukotriene levels, an altered ratio of leukotrienes to PGE2, and things that other people have shown about that population of high exhaled nitric oxide, high 8-isoprostanes, and a low lipoxin A4 level. So the last point, and maybe I'll pause for a second. Are there questions about that before I move on to the secreted PLA2 story?
Neil, are there correlation between your results and simply degree of atopy, especially perennial atopy?
Oh, great question. I probably should have brought that to the allergy group. But we actually skin tested everyone in that bronchoscopy study, and there was actually no real differences in atopy between the EIB positive and EIB negative group. The only thing was, I think it was Cat Dander was more in the EIB positive patients, but we were also testing quite a few things, so I don't know. I've always kind of considered cats to be sort of evil because I'm horribly allergic to them myself.
That's right. I mean, what's intriguing is about 25 years ago, Steve Holgate and actually people in our group showed that high dose like terfenadine could block half of EIB, which certainly fits with a mast cell component.
Right.
It's interesting it was only half.
Yeah. Yeah.
So like 240 to 360 of terfenadine.
Yeah, exactly.
In the EIB negative group, did you look to see if they had exercise-induced reflux, to see if that can explain their triggering instead of...
We didn't look at other features of the EIB negative group. You should mention that to Steve Tillis because we're currently applying for the Lung Association's airway disease. I think I sent out an email about that. But we're trying to consider what would be the important clinical question there about those patients that seem to have exercise-related symptoms and don't have a positive test for EIB. And so a lot of it could be reflux and vocal cord dysfunction and a variety of things. There's actually not really been a good population-based study, so maybe that being part of the Lung Association network would allow us to ask those bigger questions. So.
Any evidence for local division of mast cells to increase the numbers? I was thinking back in our first studies in our asthma model in mice, in the chi, we had some pictures of mast cells dividing in the airways.
Yeah, that's a key question. I think that this is I've been constructing a grant on mast cells that I'm going to be submitting, and actually one of the entire aims is about mast cell progenitors. And so mast cells have a distinct progenitor. It's actually distinct from myeloid progenitors. So like eosinophils, all the stuff that Judah Denberg showed, that in the bone marrow that you get, you do allergen challenge and you get proliferation of eosinophil precursors in the bone marrow, and those actually mature in the marrow and then are released into the circulation and trafficked to the airways. Mast cells are kind of an enigma because they don't mature in the bone marrow, and you can identify progenitors, CD34 progenitors, in the blood that have mast cell proliferation potential. And you can also, the studies from Judah Denberg and Severity and so forth, they actually also show that there's CD34 cells in the airways, so there's progenitors in the airways. And the thing that no one has ever really asked is whether or not the number of mast cell progenitors in the blood is increased in asthma, whether or not those progenitors have increased proliferation potential, and whether or not there's more of them in the airways where they might proliferate in situ. And so that's actually one of the aims of that grant. So that's a great question.
You ever come across a gene, A20, in your gene profiling?
A20?
Yeah.
I think so, but sometimes these things have different names. We can always look it up sometime. All right, so I'm going to try to go over the secreted PLA2 story here very quickly. So, when you're exercising, just mechanistically, what's happening is that you really have transfer of heat and water from the airways. The predominant mechanism of that is just water transfer, as shown in one of Sandy Anderson's studies, showing that if you experimentally induced bronchoconstriction, the amount of water loss from the airways is tightly correlated with how much bronchoconstriction you get. Of course, you have to be one of those modest number of asthmatics that actually has the disorder for this kind of thing to play out. And so what you're really doing is that when you're breathing a lot, you have water loss, and that transiently causes the airway osmolarity to increase, shown by those little dots. And mostly with the more modern models of the airway epithelium, this causes a loss of the height of the airway surface liquid as opposed to these other changes strictly in osmolarity. And the thing about it is that osmotic change occurs very transiently because the airway epithelium will rapidly restore the osmolarity. But you do have a loss of height, and so that's one thing. And then you can create the same condition by adding a hyperosmolar stimulus, such as with mannitol, and you get the same thing. And so we showed, in this same paper, when we were trying to look at what the whole regulation of this process is, that if you take, in this case, mouse lung and treat it with higher concentrations of an osmotic stimulus, you get the release of what's called interleukin 33. That's actually using a reporter assay for the receptor for IL-33. So you get osmotic stress causes epithelial release of IL-33. If you allergen challenge the mice first, before you isolate the epithelial release of IL-33, you get a marked increase in this response. And so, the one thing that we don't really have, sort of the other big thing is that, in mast cells degranulate when you exercise challenge people, as Frank was just saying, and that's actually from the original demonstration of that is actually from our study that we did when I was junior faculty here, showing histamine and tryptase release in the airway. So this is from our Blue Journal paper. But why leukocytes are activated in this in the airways is not entirely clear, and it must be some signal from the epithelium. And IL-33 is just one of those signals. You also get eosinophil activation. This was shown by Tim Mickleborough's study following exercise. And you get this kind of sustained release of eicosanoids, including leukotrienes, as well as PGP2, which is predominantly from mast cells. And so this made us very interested, in addition to the fact that that patient population has high leukotriene levels, about what's regulating this. And the eicosanoid cascade is very much regulated by the enzyme that initiates this cascade, which is phospholipase A2, that releases free fatty acids from membrane phospholipids. And that, depending on what context, can go down into both kind of pro-inflammatory and anti-inflammatory lipid mediators. And so secreted PLA2, say, release free fatty acids from a membrane phospholipid like that. So that free fatty acid, if you're especially eating a high omega-6 diet, would potentially be arachidonic acid, and that can lead down to the prostaglandins and leukotrienes. If you have the same fatty acid that's omega-3 substituted, it actually goes down the same pathway, but it doesn't make the inflammatory eicosanoids. I think the other thing in this area that's really important is that the other product of this phospholipid cleavage is lysophospholipids, and those are shown to be key regulators of inflammation. So there's 10 secreted PLA2s. These are kind of novel, and we conducted this study back in 2007. This was really a product of our work with Bill Henderson and Mike Gelb and others, where we showed that this one particular secreted PLA2 seemed to be elevated in the airways. And in this case, we screened across all of them. And then we subsequently looked at that one particular protein in severe asthma, working with Sally Wenzel, and we showed that this secreted PLA2 was increased in asthma, and so it was predominantly expressed in the airway epithelium. So we then moved on to do a definitive study here, looking at these same different groups, and in this case, we looked at the protein level in the airways, and what you can see is that the protein level for this secreted PLA2 are very clearly elevated in asthma, but in this case, it's kind of elevated in both groups of patients with asthma, not just the EIB positive patients. And like we'd seen in Sally's paper with us, that is predominantly expressed in the epithelium. And just here's some immunostaining. We did do a stereology assessment of the immunostaining, and it wasn't as clearly increased, but if you look at the relationship between the staining for this particular enzyme in the epithelium and how much airway hyperresponsiveness you have, it's a fairly good relationship, although nothing like our mast cell story. So, the kind of model, like we were saying, that there's this epithelial leukocyte connection. There's something that's happening in the epithelium, it's activating leukocytes. This seemed to be one of the key targets, and so we looked at how this secreted PLA2 could activate eosinophils, and if you just take eosinophils in culture and treat them with this enzyme, they rapidly release arachidonic acid, the precursor to the eicosanoids. They make cystinyl-leukotrienes fairly quickly, within a few minutes, and that is dose-related. And we showed in this paper really what the mechanism of that is, kind of signaling through the MAP kinase cascade. It actually looks like a good portion of it can be replicated, not by just the release of arachidonic acid as shown here, but that you release lysophospholipids, and then that further activates cytosolic PLA2. So you have both the secreted PLA2 and the cytosolic PLA2 acting together. We recently looked to see whether or not following exercise challenge you actually can see what we're proposing here, that you get increased phospholipid hydrolysis in the airways. And we found something very interesting. We haven't published this yet, but this is looking at the large aggregate to small aggregate ratio. And this is kind of a general marker of phospholipid hydrolysis. And what you see is that the patients with EIB actually have more ongoing phospholipid hydrolysis at baseline. That's why it's lower in both. We did it both in induced sputum and BAL. And then we challenged these patients. This is one of the few studies where we actually challenged them with exercise and did a bronchoscopy post-exercise, something that turned out to be pretty challenging to implement. Which is why our sample size is relatively small. But as you can see, actually both groups of patients have evidence of phospholipid hydrolysis, so that ratio going down like that. And it's just more severe in the group that has EIB, which is kind of consistent with what we saw in the cross-sectional study. So just kind of this disease model that, you get exercise-induced water loss, probably in normal people that don't have leukocyte infiltration, that causes you to release prostaglandin E2 in the airways, which is kind of a protective eicosanoid. But if you have all these ulcerations in the airways, then you make instead leukotrienes and PGD2 from leukocytes. And those have a tendency to activate the sensory nerves in the airways, which are shown down below there, and activate smooth muscle constriction. And just to show you that this actually plays out, we looked back at our samples from our Blue Journal paper and measured the leukotrienes to PGE2 ratio, and you can see that it goes up very uniformly in the EIB positive group there, and then in the normal subjects, you see that ratio actually goes down between pre and post exercise, so showing that there's this very clear relationship. So I'm just going to finish with a couple of our ongoing studies that we're doing in the lab. As after all these years, I've actually started doing some mouse modeling. It's kind of funny because, of course, my mentor is a mouse modeler, and I'm mostly held to doing human studies. But as we kind of have moved along in this area, we were interested in trying to understand the function, I guess, a lot more. We do a lot of things really in cell culture, and so this is just a couple of things though that we've done in mouse models. And so Bill Henderson had shown that this particular enzyme, secreted PLA2 group 10, plays a key role in a model of OVA-induced airway hyperresponsiveness. That's a model, and that's a very T-cell dependent model in which you give an adjuvant alum. And so it seems to, that model particularly, I don't know if you want to comment on this at all, Bill, but it sort of bypasses a lot of the innate cell mechanisms that we think are very important in some of these proteolytic allergen models. But in that model, however, the mice in which you removed the PLA2 group 10 were markedly protected, and they had an alteration in particularly type 2 cytokines in the airways and an alteration in airway hyperresponsiveness. So we've been interested in how innate mechanisms play out using this same enzyme, and so here we used a dust mite model in which we gave initial sensitization dose of house dust mite in the airways, and then we followed that by airway challenge with house dust mite. We did this in a mouse, a black 6 mouse that has knockout of the group 10, PLA2 group 10, versus the wild type. And it's very interesting. A lot of people in their dust mite models don't get airway hyperresponsiveness, which is shown here, a methylcholine challenge. And so we spent really like a good year and a half trying to work out our model so that it looked like we could look at that aspect of asthma. And so our dust mite model, they get hyperresponsive. And you can see that the group 10 knockout mouse is protected from airway hyperresponsiveness there. They also have a marked reduction in the number of airway eosinophils, T cells, and macrophages there in the airways. But I think, and they have a reduction in total IgE. I don't have it up here, but they actually have a reduction in dust mite specific IgG. We've still been having trouble measuring the dust mite specific IgE level, mostly because there's so much IgG against dust mite in the samples. But so it seems like it has an alteration in adaptive immunity, which is very consistent with what you usually see in the OVA model. But there's something else here that's really interesting. These mice have less interleukin 33 in their airways, and this is a really very crisp finding. And so one of the mechanisms is really that is through these innate lymphoid cells. And so what we can see at the bottom is that innate lymphoid cell type 2s are increased in the airways, and in the knockout there's a reduction in those innate lymphoid cells. So we've been mostly lately trying to solidify these results using a cell culture model about how epithelial PLA2 group 10 regulates interleukin 33, which seems very interesting. But the second model we've been using is really a much more novel thing, and it would take me a while to really explain what made us think about this. But really the idea that PLA2s can act as adjuvants directly. So when you're using a model like the OVA alum model, you're using the adjuvant alum, which is this kind of uric acid dependent adjuvant in the peritoneum. But there was this bee venom, the study on bee venom anaphylaxis that was in the journal Immunology that made us think that PLA2s could actually serve as an effective adjuvant. And no one had ever challenged the animals in the lung. This was just this one study. It was done by the group in Yale, and it was in Immunology, which is one of the top journals. So we created a model in which we essentially set up the same model as the OVA alum model, except we used the PLA2s as adjuvants. So in the peritoneum, we sensitized the mice by giving them PLA2 group 10 plus OVA, or a catalytically inactive mutant of PLA2 group 10, or no adjuvant, and then we followed that with OVA challenge. And so interestingly enough, we actually did this initially with bee venom PLA2, and we got the same results. So we have increased airway hyperresponsiveness in both the catalytically active and the catalytically inactive enzyme there. So increased airway hyperresponsiveness, but it doesn't appear to use the catalytic activity of the enzyme. But then if you look at the trafficking in type 2 type findings, so eosinophils, dendritic cells, T cells in the airways, it's only in the catalytically active adjuvant, as shown there. This also tracks very clearly with OVA-specific IgE and OVA-specific IgE. So the adaptive wing of this is all being through the catalytically active enzyme, but there's also something about airway hyperresponsiveness that's discordant to that. And then if we take lung leukocytes out, so these are lung cells that are re-challenged with OVA, and you can see, and this is true for a number of type 2 cytokines, that there's an antigen-specific increase in IL-13, so OVA-induced IL-13. That doesn't occur. So there's a very interesting thing in that the catalytically active version of PLA2 group 10 seems to be important in, as an adjuvant with the adaptive immune response. There's also something about the catalytically inactive version of this, and maybe that's through binding directly to a receptor that binds to the PLA2, so it's very interesting. So anyway, I'm going to just conclude with that about EIB being a distinct feature of asthma. It's related to mast cell infiltration. Th2 high seems to track along with indirect hyperresponsiveness and mast cell infiltration. And as I touched on at the end, epithelial-derived secreted PLA2s are probably a key regulator of inflammation and lipid mediator formation. And I'll just end with acknowledgements from my lab, and thanks for your attention. The inhibitors for group 10?
Yeah.
Well, yeah, interestingly enough, actually Bill and Mike, they put in the human PLA2 group 10 into a mouse so they could use an inhibitor because they don't have an inhibitor for the mouse PLA2 group 10. And so in that OVA-alum model, that showed that well, first that the human group 10 could mediate airway hyperresponsiveness and that the inhibitor was effective, the human inhibitor.